very much for the introduction, Hudson. Thanks also for the opportunity to talk to you about our rare disease research. And as you've rightly pointed out, I do see patients with rare diseases, and unsurprisingly, my research also focuses on individuals affected by rare disease. So I thought I'd talk a little bit about some of the challenges and opportunities that we've faced in this area that we've been looking at for a number of years now, and just sort of to highlight a few things that we've encountered in rare disease research. First thing to say really is that there are many rare diseases numbering in the thousands, and individually these rare diseases affect, by definition, fewer than 1 in 2,000 people. However, collectively rare diseases affect about 1 in 12 Australians. These conditions are usually really difficult to diagnose, and they often take somewhere between 5 to 10 years to actually get a precise diagnosis. And along the way, patients are often misdiagnosed or go undiagnosed for many years. And even when diagnosed, they often don't have any particular specific approved therapy or treatment. So central to the management of any individual with a rare disease is the diagnosis of the rare disease. And as part of that workup, often there is clinical evaluation, lots of investigations, including genomic testing. Once, if all of that evaluation and genomic testing does arrive at a diagnosis, that then opens up for the individual potentially specific clinical management guidelines, but also potentially things such as clinical trials or therapies. And of course, for the family with genetic conditions, it can also inform reproductive management of the parents of the affected child or other family members who are potentially concerned about having an affected child themselves. Once a diagnosis is made of a rare disease, that opens up research possibilities such as natural history studies, qualitative studies such as quality of life, and also clinical trials and therapies for rare diseases. Now, just briefly, I think it's worth talking about genomics and the genome and what we mean when we talk about genetic and genomic testing. We know that the human genome is organized into multiple levels. So it's a little bit like zooming in and out of Google Maps. So if you zoom all the way out of Google Maps, you can see all of the continents. It's similar to looking at chromosomes under a microscope. And so that's at the very macro level, looking at the entire human genome. However, you can scroll right in and you can zoom right into the individual chromosome and zoom in even further right down to the individual gene, or even at the single nucleotide level for DNA. We have tests that are available for all levels of the genome, and they're sort of listed on the right there. But essentially, any level of the genome can be interrogated with specific genetic tests. What we want to focus on today, I guess, is and what we've been doing, particularly since the explosion and the revolutionary change that genomics has brought, has really been changing in clinical practice. And with genomics, instead of looking at individual single genes, genomics brings the power of being able to look at multiple genes at once, and potentially even all 20,000 genes in the human genome. Not only that, we also have exome sequencing. We also have whole genome sequencing, which is akin to looking at the entire orange, whereas the exome is like looking at just a slice of that orange. So really what genomic testing is all about is looking at the DNA strand and reading the DNA strand and reading individual genes and sections of DNA. And exome sequencing is really focusing on the exons or the protein coding regions of the gene, whilst the genome sequencing will cover all of the bits that are beyond the exons and looking at the whole orange instead of just a slice. There are also, so when we're talking about genomic tests, we can talk about doing a very specific panel, so looking at a very set list of genes, We can be looking at the exome, which is looking at about 1 to 2% of the protein coding regions of the genome. And finally, we can look at the whole genome and sequence everything, including exons and introns. So this is hopefully just to lay the foundation of what we're talking about when we talk about genomics and rare disease research, because it is genomics that has really revolutionized what we can do in the diagnosis of individuals affected by rare disease. The other important concept to think about when we are looking at rare disease research, and this is an important distinction because it's not often appreciated that there is a distinction between clinical testing versus research testing. Now, clinical testing is very much like a Qantas flight, which I'm going to catch in about an hour and a half. This is really about having a very safe, standardized procedure that you can base medical decision making on. So this is applied in the clinical service. The same rules, I mean, similar rules apply for consent. Obviously, you have to have informed consent to proceed with clinical testing. But clinical testing relies on looking at very set gene lists with genes that have an established gene disease association. There are, of course, some ethical issues and some potential issues that we often discuss with parents when we consent them in the clinical service to talk about things such as incidental findings, that is, genomic findings that are off target, that are not actually related to the child or individual's primary condition. And of course, if we are doing trio exome sequencing or family-based sequencing, when we are looking at the child's genomic results, we're also adding in both parents, both biological parents, for that sequencing. And there are sometimes the uncovering of unexpected family relationships. And that's often related to, not often, but when we talk about that, that's usually in relation to alternate paternity. So where the father or the stated father is not necessarily the biological father. And we'll talk about that in a second. And of course, with clinical testing, we also have to talk about data sharing with families, because any genomic results arising from that child or their parents could potentially be relevant to other family members. We also talk about data sharing in the context of global databases, and I'll drill down on that a little bit further. The flip side of clinical testing is research testing. Now, research testing is usually a slightly different context. So whereas clinical testing is done in a NATO accredited laboratory, research testing is usually done in a non-accredited laboratory and in a research lab. And that's usually done within the context of an ethically approved research project. And it comes, of course, with informed consent. It's a bit like the fighter jets now. You've now got the freedom to look at all genes, not just the genes that currently have a known disease association. Just to give you some context also, out of our 20,000 genes in the human genome, roughly about 4,500 are currently associated with human disease. The other 15,500 genes, we don't really know what they do, and they're not currently associated with any human disease. Of course, with research testing, we also have the similar issues like incidental findings or the uncovering of unexpected family relationships. But often with research testing, if a patient has already had clinical testing, a lot of those things will have already been identified through the clinical service and already been managed. So research testing, we usually see less of that in the research environment. We also have things to consider like data sharing, similar issues like data sharing with global databases. And additionally, we have the additional issue of tissue sample collection with research analysis. And I'll cover that again briefly. So just to give you an example of some of the research that we're doing in rare disease research, we have what's called the RDNow research program, RD standing for rare disease. So the Rare Disease Now research program, wherein patients are recruited after having had a clinical genomic test. So this is an important distinction again. So patients have already been seen in the clinical service. They've had a genomic test, most often an exome sequencing. And from that, about 50% of individuals remain undiagnosed. So they haven't got a diagnosis after clinical testing. They are then enrolled in the RDNow research program to then undergo further analysis in order to either discover novel genes, mechanisms, and treatment, and potentially to then look at essentially identifying a precise diagnosis for them in order for them to have appropriate care and to then potentially lead on to available therapy like and clinical trials if available. So this is essentially just a snapshot of our rare disease research program, and it's the key point is that these individuals who have already had clinical genomic sequencing and they are undiagnosed and they come through a research program. There are some challenges, obviously, with any rare disease research and participants, mainly grouped into recruitment, consent, and retention. With recruitment with rare diseases, patients are often underdiagnosed or under-ascertained, and it can be difficult to reach populations of individuals with rare diseases. And this can, of course, can affect equity of access. And this is also a problem with diagnostic codes that we have. So our electronic medical records, for instance, at least in our centre, does not capture a lot of rare diseases. So a lot of individuals in electronic medical records are not coded, and if you're not coded adequately, you can't be identified with a particular rare condition. And even our sort of global rare disease or global ICD codes, for instance, do not cover rare diseases adequately. Then we have the issues of consent and management of consent, particularly in individuals who are of culturally and linguistically diverse backgrounds, or First Nations individuals where there might be variable levels of health literacy. And of course, that can affect, again, equity of access and challenges with explaining the research program and explaining all of the different ethical challenges associated with that. And of course, when you've got a rare disease research program, there's often a fairly long diagnostic trajectory. And so sometimes over the course of a program, things that we want to do in that program might change. And so consent management is important to consider there. Data storage and sharing is always something that we have to discuss with families as well, because that is something that we need to do as part of a research program in order to connect with other individuals with rare diseases. And of course, when you've got a program that runs over a long period of time, you often get, you will get patients who then become lost to follow-up, which can then jeopardize the quality significance of your research and follow-up studies. And you can lose that longitudinal data that is so important for natural history studies, for instance. So some of the challenges that I want to focus on a little bit further that I've already touched on included the issue of incidental findings, which is something that a lot of people are concerned about or worried about, but in reality, it does not actually happen that often. We find that incidental findings, so these are diagnoses or findings in genomic tests that arise from genomic testing that are not actually related to the primary reason why that child or that individual has come to clinic. And this maybe happens about 1 to 2% of all clinical tests. And these usually arise, these diagnoses might come about for instance, an adult onset condition in a child, or usually a carrier state. So a child is identified to be a carrier of a rare recessive genetic condition. And these are usually managed very sensibly by genetic counselors in the clinical service and managed over time through there. So by the time they come to a research program, these have already sort of fallen out and already been managed clinically through the clinical service. And they don't actually happen very often in reality. The other thing that happens more specifically in a rare disease research program is that we ask parents for the potential for collecting of tissue samples. And this is usually saliva, blood, or skin, or sort of clinically accessible tissues, and occasionally other tissues such as muscle. So these are usually collected opportunistically. Say, for example, if a child is already having an anesthetic for some other reason, such as having a brain MRI scan, or having surgery for management of their issues, we usually then request that a sample of skin be taken at that stage. And it's usually a very small biopsy and managed that way. The reason for doing this, for collecting these samples, primarily is for diagnostic purposes. So in other words, what we're doing by collecting these samples is actually wanting to get a diagnosis for this child and this family. The other purpose of getting these samples is that we might want to do functional assays. So these are tests on cells that we collect from this either from the blood or the skin or the saliva, and these are tests that can be done in the laboratory to try and confirm any genomic findings. So in other words, we might find a variant of uncertain significance in a gene that we might then want to follow up with looking at that gene in the cells and looking at the dysfunction and whether there is any dysfunction that can be seen in the cells, which would then confirm the pathogenicity of that particular variant. We might also then want to do further research analysis, such as making induced pluripotent stem cells to look further at the function of that particular gene in a more relevant tissue. So you might want to get those iPS cells from skin, derive iPS cells from skin, and differentiate them into a different tissue type, a more relevant tissue type to study that gene's function further. And again, that's to do with diagnostic purposes. And in some rare disease research programs, biobanking is important, and tissue samples are collected for that purpose. Data management is an important issue, and this is often in the clinical setting where we share data, particularly with other family members, because it might be clinically relevant for them, and most families are very happy to share genetic information with their family members. It is also standard lab practice to share any variants, information about variants and the classification about the pathogenicity of variants with global databases. But of course, this all occurs without any identifying patient details. And this is at the gene or variant level. So these are often shared so that the lab often says, well, we found a variant in this particular gene and we classified it as pathogenic or benign or of uncertain significance. And often if we do share clinical information, this is in a de-identified way with only what we use are the HPO terms. And this is the human phenotype ontology terms, where these are terms that just sort of describe the features of the patient, like intellectual disability or seizures or reflux, that kind of thing, as just specific HPO terms when we share clinical information. And of course, like I said, it's de-identified. And unexpected family relationships is something that is also something that gets raised as a concern, but this very rarely occurs and is often identified during clinical testing and managed clinically. So it's not really a major issue at the research level. We've undertaken a lot of consumer engagement and from rare disease communities. In fact, the RDNow program actually has a consumer engagement committee, and the feedback that we get from them is that patients are often very motivated to participate, and they're looking to connect with other people with rare diseases. They also see benefit in even having negative analysis on further genomic testing. They're very willing to share genomic data for the advancement of human knowledge and science. But the important issues for patients is equity of access, particularly people who are living in remote or regional remote areas, or people of First Nations or culturally and linguistically diverse backgrounds. Patients with rare diseases have often told us that they want to be part of the design or co-design the research project and engaged from day one. But they've also fed back that they find the consent process can be laborious, particularly if the consent process, if the project changes over time, and having to go back to families to ask if we can do different things at different times. and they want that to be to limit that as much as possible, but they understand, and there's a lot of work that's happening on the dynamic consent front, so that families can have a bit more agency over their consent process, and we're also looking at simplifying consent forms and looking at alternative ways to get patient consent. We've also been running focus groups with rare disease researchers, and of course, time and time again, we get that the barriers for rare disease researchers is time, funding, and ethics applications. They find that they're in siloed work research, and they want more data sharing between research groups and access to biobank samples. They value the involvement of genetic counselors precisely because of some of the ethical issues that I've already covered, that I've discussed already. A lot of rare disease researchers, particularly junior ones, want help with ethics applications. They find the consent process laborious also, and also echoing the rare disease patients, they find that having to go back to patients repeatedly is often time consuming. And again, looking at dynamic consent and long-term consent management are key issues for rare disease researchers. There's also talk about whether broad consent for all patients coming to the hospital is appropriate, or whether that can be implemented. And there's lots of work still in process about that. And of course, a lot of clinical researchers are wanting training in clinical trials. So as an example, we have a platform program. So the RDNow, we have an RDNow Umbrella platform program wherein our research program is able to sort of host other rare disease research, and any research that is aligned with our goals and purposes can come under the RDNow umbrella, and make use of the templates and legal agreements and materials transfer agreements, for instance, for research. And so we've got this platform program, and that's been useful for researchers who then can go through the ethics application. They still have to use their own, still have a defined ethics protocol and ethics consent forms, but they can make use of our templates, and so they're not reinventing the wheel every time. So I think there are some opportunities that we're looking at. We're looking at developing some streamlined consent processes, using videos and animation or web-based processes to get consent from families. And that can also be helpful for improving equity of access, because you can have subtitles and reach and get other languages in there. We're looking at ethics training, particularly for junior researchers in clinical trials. And of course, we're in discussions and collaborations with people who are developing dynamic consent. And of course, I've talked about our program-specific platform. And of course, all of this is to ensure that we are, and it goes without saying, that this our research continues to be aligned with the NHMRC statement on ethical conduct of human research. So I think this is just hopefully just a quick rundown of some of the rare disease research that we're doing here at the Royal Children's Hospital and Murdoch Children's Research Institute and Victorian Clinical Genetics Services. Happy to take any questions. Thanks.
Thank you, Professor Tan. I found particularly your opening remarks on genetics and genomics particularly clear and comprehensive. And I wish you'd taught the genetics course I took as a freshman at university, except that most of what you presented wasn't known back then. So yeah. I see we now have one question that has come in. How does dynamic consent work?
Well, that's yet to be fully worked out at this time. But dynamic consent, in principle, is that that instead of having a paper form that you sit down in front with a patient and you fill out a form in front of them, dynamic consent can be web-based or computer-based, for instance, and these can be updated, and patients can be contacted dynamically, and to get if there are any changes in the protocol or changes in the questions, and patients can change their consent over time. They can dip into their consent form and change their optional consents, for instance, for example, tissue collection or photographs. and dynamic consent can also be used for long-term consent management, and easily and updated consents can be more easily version tracked. So these are all, that's the general concept of dynamic consent, but it's not been fully implemented yet. There's lots of talk about using that, but it's not been fully implemented on the ground. It's just something that I think a lot of people are talking about and trying to implement for getting patients ease of consent and consent management.
Gordon, you have something you'd like to add to that?
Yes, of course. Thank you, Tiong. Very, very good, and great to hear about dynamic consent. And then, look, it's clearly important. So, in terms of the research and the data on people who wish to donate samples, etc., are many people likely to say no? So that's the first question. Are many people likely to say no to finding to using their samples for other purposes? Do you know?
Oh yeah, so in my experience so far in our rare disease program, we've recruited over more than 300, probably close to 400 patients now, and in my experience, very, very few have said no, we don't want any samples to be collected. That's been an extreme minority, because most patients actually appreciate, once we explain to them, look, if we are going to collect a sample, it is for diagnostic purposes. We're using it because we are trying to establish a diagnosis for your child. Secondly, if we are to collect a sample, we are doing it in an opportunistic way. So it's not like we're arranging a second general anesthetic, like another general anesthetic. We're already doing it in the context of the child already having an anesthetic or having a procedure for some other reason. So in that context, most parents have said that's totally fine, and they're very happy for that to happen.
Yeah. And a second question, if I may, Hudson. So Tiong, you of course mentioned that. You mentioned dynamic consent, and that's yet to be decided how that's going to work. But I think, as you also pointed out, it's really a better approach is to broaden consent in the first place. And the reason I say that is in legislation, for example, you can use data for purposes as long as it's going to benefit the community. And I'm not saying one should ever use samples without people's consent, but clearly having that broad approach to consent where you offer people a specific opportunity for the samples to be used for diagnosis and for research, or for both, would probably be appropriate, dynamic consent notwithstanding. So, you talked about using them for diagnosis. Do you ever go for the double of diagnosis and research at the same time, or do you consider them to be sufficiently different that you would have to have different consents for that purpose?
So I think at the moment our consent forms talk about that the samples that we when we sit at the clinical level and we're saying, when you do this genomic test, do you consent to be contacted about future ethically approved research projects? And again, the vast majority of parents will say, yes, that's totally fine. They want to be contacted. It doesn't mean that they're obliged to go forward with if they're contacted about that, but pretty much all of them say yes, they're happy to be contacted about.
I only just mentioned that. Sorry, Hudson, I'll just finish with this one. I only mentioned that because you say they don't like to be recontacted. So clearly you could do this, at the same time, and that means they don't have to be contacted again. But it just seems a bit strange to be recontacting them when they don't really want to be recontacted. Anyhow, I'll leave it there. Hudson, over to you.
And we're exploring that right now. We've actually got a research project looking at rare disease research participants and saying, looking at their appetite and to what level do they want to be contacted? How many times can we sort of recontact you? And that's an ongoing research project.
Yeah. Thank you, Tiong.
Professor Tan, what's your experience been with your local ethics committee? Have you found any barriers for the committee members, reviewers, in understanding how a platform trial works? Especially when you start adding new domains or sub-studies?
No, I think we've had a really good run with our local HREC, our RCH HREC. Obviously, they're in the audience, so I'm not going to say anything bad. But no, I'm just joking. But in all honesty, we've actually had really responsive and engaged research committee, and they've been really open to just discussions about how best to do things in an ethically, ethically sound way. So not had any major barriers. I think where the challenges come in is when junior researchers come on board and their protocols need a lot of refinement. So there's a lot of toing and froing. And so sometimes that time taken to sort of get a research protocol and project off the ground can sometimes be a bit of a challenge, because we're just trying to refine all of those processes. But we don't find that, we find that the ethics committee to be extremely generous with their time and engaged and responsive.
What are your thoughts on consumer or community involvement in research and the role of community researchers?
Yeah, well, I think that's really been important. As I said, since day one of our research program, we've had a consumer engagement committee comprising multiple representatives from multiple rare disease groups involved, and having their input into the design of a project and the implementation of a project, and looking at ways in which the direction and the findings and the outcomes of the project are going to benefit the rare disease communities, I think has been super important for us.
Thank you. Thank you for your talk. And